Hello, my name is Nathan Friesen, and I would like to welcome you to this presentation on phenocopies. This presentation is based on a book chapter titled, What Does the Phenocopy Copy? Originals and Fakes in Biology, which was written by Susan Oyama. My partner, Dawson Doucette, and I hope that the next 30 minutes or so are an interesting and useful exploration of this fascinating concept in developmental biology, which has wide-ranging implications for evolutionary theory in general. Without further ado, let's dive in and learn about phenocopies. To start out, we're going to define a few terms that will be important to understand this presentation. Firstly, we offer a definition of phenocopy from the man who coined the term. Goldschmidt, all the way back in 1929, defined a phenocopy as an environmentally induced character which closely resembles a known mutant. That is, a wild-type genotype which is induced to exhibit the phenotype of a known mutant through environmental effects. While we will see throughout this presentation that this may be an oversimplification of the phenomenon, it is an extremely useful working definition to begin to explore the phenomenon of phenocopies. Next, we will define phenylketonuria, which is a genetic condition wherein the patient's body cannot properly break down phenylalanine, which is an essential amino acid which must be acquired through one's diet. However, in the body of phenylketonuria patients, their bodies are unable to properly metabolize phenylalanine, and the excess buildup of phenylalanine in the body will result in developmental difficulties, including severely reduced cognitive function. Thankfully, it is possible to avoid these deleterious effects through the implementation of a strict phenylalanine-reduced diet. Lastly, the term epigenetics is used to refer to modifications to DNA that don't change the actual base sequence, which are used to control or change the transcription of target genes. While this can be done through a number of methods, some of the most common are histone modification and methylation. This talk will begin with a discussion of the terms genotype and phenotype and how they have been understood in the past, and why phenocopying can make the distinction and relationship between the two murkier than it would seem at first. It is not as easy to draw a direct line from genotype to phenotype as it may have once seemed. Diving deeper into this idea, we will look at the false dichotomy often presented between nature and nurture sources of variation in development. Rather than seeing the two forces as opposite binary forces, it is imperative to see them as intrinsically linked and influencing each other in a reciprocal manner. Thirdly, we will look to recast developmental pathways in a new light as less certain, predictable paths from genotype to phenotype. We will see that it is inaccurate to view development as a straight path from one point to another, but rather we will see that development is a highly indeterminate process with a high degree of plasticity in both the paths that it can take to its endpoint and the actual endpoints it reaches in adult organisms. This plasticity is thanks in large part to the importance of gene-environment interactions and how each can influence the other. After that, we will look at some examples of environmental impacts on phenotypes in nature. We will use these to illustrate how it can sometimes be difficult to clearly classify whether some particular phenotype is a phenocopy or the natural product of a particular set of gene-environment interactions throughout development. After that, we will take a closer look at two different styles of environmental influences in phenocopying, which can be through intentional or unintentional phenocopying. Next, we will look at a particular example in bacteria that the author used to show how defining what is a phenocopy and what is a model phenotype can be extremely difficult in some circumstances. Often, in real life, the categorization of living things is not as cut and dry as theory might suggest. We will finish this talk off by looking at some current questions about phenocopying, which warrant more investigation and experimentation to see where we should go from here.
Firstly, let's define what phenotype and genotype are. In general terms, genotype is the actual genetic makeup or material of an organism. That includes its sequences of DNA, or what we also like to refer to as genes. These are the specific instructions for a specific organism. Now, on contrary, phenotype is the appearance of these genetic material when expressed. That is the character traits that result from the expressed DNA or expressed genes. Now, genotype and phenotype have often been, known as a dichotomy that is polar, meaning that genotype and phenotype are separate. In most cases, it was often thought that the genotype would code for a specific phenotype. But with what we now understand about phenocopying, this is not always the case. Phenocopying now is an environmentally induced character which closely resembles a known genetic mutant. So in other words, the genotype does not always show what the phenotype is. When environmental factors play on a genotype, a different phenotype may arise. So the current definitions of phenotype and genotype don't actually stand when phenocopying is understood. When we often think about phenocopying, we are thinking of how that changes the original definition of genotype and phenotype. No longer is it genotype produces phenotype, but that it's more to that. Phenocopying refers to environmentally induced characteristics, so that there is an environmental influence on the genotype to produce a specific phenotype, dependent on what those environmental factors are. Now, this kind of connects to the idea of another dichotomy, which was nurture versus nature. Nature being the sole matter of an organism or the physical thing itself, the instincts that it may have had, where nurture being the environment it grew up in, the influences it had from other organisms. But phenocopying no longer has this separate dichotomy of nurture versus nature, which one was more important. But it's nurture and nature, and that they're kind of interconnected. A phenotype can be thought of as maybe given by nature, in the sense that nature is the genes or the material gives a specific phenotype, it produces it. And the nurture part of it is fundamental and inherent with the nature. So it's connected. There's an interplay between the genetic material and the environment, which produces a phenotype. No longer are they separate entities, nurture separate from nature, or genotype separate from phenotype, but that they are connected.
Implicit in the idea of phenocopies is the fact that genotypes have multiple potential developmental pathways. There is no single pathway which a genotype must follow to its phenotypic endpoint. This plasticity means that a particular phenotype, normal or mutant, may often be produced either through standard genotypes and abnormal environmental conditions, or it may be produced through standard environmental conditions with an abnormal genotype. It is important to note that phenocopies imply that long-term developmental predictability is impossible because continuity in development is environmentally dependent, and a perturbation in the environment can easily switch a genotype from one developmental pathway to any other of its possible developmental pathways. In this way, we can see that a model of development which draws a straight line between any particular genotype and a corresponding phenotype is not quite accurate. Rather, we should see that genes and environments interact and collaborate to produce any of the possible phenotypes for any particular genotype. One illustration of the developmental plasticity inherent to nearly all genotypes is seen through a closer study of phenylketonuria, which we defined earlier, and is a genetic disease which involves an inability to break down phenylalanine in the body. While it has a strong genetic component, environmental modifications can result in a normal phenotype even with a mutant genotype. Thus, the phenotype of reduced cognitive function cannot be attributed exclusively to genetic or environmental factors. Rather, the gene and environment are interacting in a phenylketonuria patient to produce the severe reductions in cognitive function that are hallmarks of the disease. Importantly, this shows that developmental pathways cannot be clearly predicted or assumed because the environment is highly variable, and if the environment changes, the developmental pathway can be switched and a new phenotype may be the result. This is because each genotype does not represent one corresponding possible phenotype, but a whole range of phenotypes, which may include amongst them mutant and normal phenotypes. One more important way that genotype and environment can interact is through the effect that genotype can have on an organism's developmental environment. In fact, the actual environment of a developing organism is often partially created by and somewhat dependent upon its genotype. One clear example of genotype influencing environment is present in the many different morphogenetic gradients which are present during development and cause some genes to activate or deactivate. While it is often not thought of as the environment of an organism, the environment in which a cell resides is of utmost importance to its relative expression of genetic information and genetic products, like proteins. So this level of environmental influence cannot be ignored and is often heavily influenced by an organism's genotype. The picture on the right here is an example of this. It shows, in just a simple image, the reciprocal gradients present in the developing embryonic heart of proteins sonic hedgehog and bone morphogenic protein and Wnt protein. Together, these three proteins, which are themselves produced by cells in the embryo and thus produced genetically, define the developmental environment of the cells which will eventually become the heart. And it is through these proteins' interferences and interactions that highly specific localization of heart cells can begin in the developing heart. Clearly, the genotype of the organism has in this case influenced the environment of the organism, and when this goes wrong, many developmental abnormalities are possible. Next, we will talk about some ways in which environmental conditions can influence epigenetics in an organism. We will look at two examples where this has been shown to produce heritable changes to the epigenome of an organism. First, looking at Drosophila, we can see an example of a heritable epigenome change in flies who developed red eyes when raised at 37 degrees Celsius. These flies were from pure breeding white eye lines. That is to say, that their genotype would normally have developed into white-eyed flies. However, with the right environmental interactions, in this case, a raised temperature during development, this genotype was switched to a developmental pathway resulting in the exhibition of red eyes. Fascinatingly, these red-eyed flies passed on this epigenetic change to their offspring, who also developed red eyes even when raised at a standard temperature. The environmental effect on the parental flies had imprinted on their epigenome, which resulted in offspring with red eyes, even though they carried the white eye genotype. Another example of this comes from the Dutch famine of 1944 to 45, where after an extended period of undernutrition, women gave birth to children who experienced higher risks of a myriad of diseases and were also likely to be born smaller than normal. Most interestingly, however, when these children, who grew up without experiencing famine, had children of their own, those children were also smaller than normal as a result of lasting epigenetic changes from their grandparents' experience in the famine from decades before.
When we're thinking about phenocopying, there are primarily two different styles of environmental influences that can affect a phenocopy. First off is what we know as intentional environmental influences. And these are when scientists purposely introduce a altered or a differing environment or environmental factor or treatment onto a developing organism to environmentally induce a characteristic that is not typically seen from the genetic makeup of that organism in normal conditions. Unintentional environmental influence is that seen in the wild or in nature that naturally occurs. An example of this, as seen on the slides, is the Himalayan white rabbit. The Himalayan white rabbit in the wintertime has black fur on its feet, its ears, its nose, and its tail. These are important extremities that usually need to retain a lot of heat, and so in the wintertime, they have a black fur, black pigment kind of develops on those extremities. However, when the environment changes naturally, as it shifts to summer, as you can see in the picture on the left, the rabbit loses a little bit of that black fur in its ears, its nose, and its tail, as it does not need as much heat, absorption in those areas in the summertime as the climate is different. And so this is an example of an unintentional environmental influence in which the genotype of the rabbit stays the same, but the phenotype changes, that being the color of its extremities, as the environment changes as well. Let's look at an example of an intentional and an unintentional environmental influence and how it relates to phenocopying. One example of an intentional environmental influence can be seen in experiments done on a chicken. Rumplessness in a chicken, which means the absence of the tail feathers, occurs when insulin is introduced in very early development. And when it is introduced in late development, we see a shorter upper beak emerges. So this is an example of a scientific experiment in which insulin was given, something that is very abnormal for chickens. And when it was administered in very early development, we see a very abnormal phenotype change. Although the genetic material of the chicken remains constant, the phenotype is changed based on an external treatment or an external environmental influence that was intentionally provided to this chicken. This does not occur naturally in the wild, and so we see a very abnormal phenotype that is produced when insulin is introduced to a chicken. On the other hand, an environmental influence that is unintentional is the genetic condition known as ocolema. This is the development of abnormal arms and legs, as the arms and forearms and thighs and legs are absent, and the hands and feet sprout directly from the trunk of the body. This condition occurs in two ways. First off, it can be a result of an abnormal genotype in which random genotype mutations occur. And the second cause is with the presence of thalidomide during prenatal development. Now, thalidomide was a treatment used for morning sickness for pregnant women and has since then been banned because of the results of thalidomide in prenatal development can cause focolema. Again, this is an unintentional environmental influence where either naturally random gene mutations occur, or thalidomide, which at the time was not known to cause or was used to treat morning sickness, had an impact on the development of prenatal babies. I'm going to shine light now on an example that the text, the article used about phenocopying in bacteria. Now, bacteria comes in all different shapes and sizes and colors, and it can be a really good way to study phenocopying. For instance, in this example, we see that bacteria strain 1, which is a bacteria that grows on a specific medium, is the color white. And when we look at bacteria strain 2, which is a related bacteria, it grows on the same medium as bacteria strain 1 and is yellow. However, when we take bacteria strain 1 and we grow it on a different medium, we can see that phenotypically it can be turned the color yellow. So here we have an example of phenocopying, where bacteria strain 1 is environmentally determined based on the medium in which it grows on, where bacteria strain 2 is genetically determined as it grows on the same medium that bacteria strain 1 was the color white. Bacteria strain 2 is the color yellow. But when we see bacteria strain 1 on a different medium, we get a environmentally determined phenotype which mimics bacteria strain 2. So in this scenario, which one is the phenocopy? And the phenocopy would be bacteria strain 1 because its environment dictates its phenotype. But what if I proposed a different question? What if the white bacteria can also be a phenocopy? In this example, we see that there is a bacteria strain 3, which is unknown and contains a different phenotype than bacteria strain 1. However, when this bacteria strain 3 is grown in a different medium, it can phenotypically mimic bacteria strain 1. In this example, bacteria strain 3 is an environmentally determined phenotype which mimics the original bacteria strain 1 in its normal environment when it is white. So this poses the question of, in this scenario, which one is the phenocopy? For this instance, bacteria strain 3 that has been grown in a different medium, that is to mimic or phenocopy bacteria strain 1, would be the phenocopy. But we just said before that bacteria strain 1, when grown in a different medium, was the phenocopy. So how do we answer this question? And the answer to this question is that both can be phenocopying forms. This is where phenocopying has its limitations in what exactly phenocopying means. Bacteria strain 1 can be phenocopied to mimic bacteria strain 2, but there might as well be a bacteria strain 3 that can be phenocopying bacteria strain 1. So how do we know what exactly is the right exact strain? It is all dependent on the environment in which the bacteria grows in. And that's just it. The difference between all the bacteria is that they're environmentally dependent because it is an alternate medium that is required to produce the phenocopy. There is no one right medium in which the bacteria can grow, and so the phenocopying can only occur when different mediums are, provided. In that sense, that it's the environment that the bacteria grows in that determines either it's a phenocopying form, if it's a phenocopying model, or if it is genetically induced. How do we to say which one is which? And this brings me to my last slide, and that is questions that we still need answers to about phenocopying. The article title is, "What does the phenocopy copy?" And I guess the short answer to that is that phenocopies do copy a phenotype, but who's to say which is the original and which is the imitation? In the bacteria example, we saw how the environment dictated what color the bacteria was, but who's to say which is the correct environment for the bacteria to grow in? Another question to answer is, do each copy each other, or is there specific environmental conditions? And how do we define which is the normal environment for development? We can add external factors, right, treatments to development, but who's to say which environment occurred? How do we not know that thousands of years ago that the environment was so different, in which different chemical components could have influenced evolutionary processes in phenotype and phenocopying in that sense? Another question to ask is, does phenocopying only apply to early development treatments, or is it still counted in later life treatments? In the example of the chicken, we saw how phenocopying occurred when we introduced insulin at a very early development in treatment, but later on in treatment we saw, or when insulin was introduced, it had a different effect. It caused a different growth in the chicken's beak. But we can also think of phenocopying as, if we're changing the environment later on in life, I guess different from an abnormal, we can think of the same thing with genetic diseases. When we are providing treatments or medicines to treat those genetic diseases later on in life, does that count as phenocopying? Technically we are inducing a different environment from the normal to get a different result or a different phenotype that is expressed. So another question to ask is, what counts for phenocopying? Is it only applied to early development treatments, or does that still count in later life treatments as well? Another question to be asked, or I guess to answer as well, is, if the environment plays such a great role on phenotype expression, are we to rethink the dynamic possibilities of the cell and its genes? Previously, we think of the cell as a energy containing device, but Klopfer, a scientist referenced in the article, thinks that we should be thinking of cells as information generating devices and not necessarily information containing devices. And this is because they produce compounds dependent on the immediate environment. Cells use external environment factors to determine what in fact they code for. And so not only do they just contain the genotype, but that the external environment has a factor on what phenotype is expressed. And so the way we typically think about cells as these DNA containing devices are maybe potentially more supposed to be thought of as information generating devices, in which the environment has a greater impact on which pathways happen, and in that case, what phenotype occurs as a result to a changing environment. Briefly here, these are some of the sources that we used for either images or information in this presentation. I hope you enjoyed it. Thank you for listening.